All right, so can you all see my screen? We're in active transport. I know we have a lot of slides to get through, but we're going to try to get through these today. We were talking about passive transport last time, and passive transport does not require energy at all. Active transport, in contrast to that, does. And when we say energy, we mean ATP. So active transport, it's a carrier-mediated process requiring ATP, or energy, and it moves substances across the cell membrane. Remember, we're talking about the cell, and we're looking at the cell membrane and how things get into and out of the cell. Active transport will always move substances from regions of low concentration to high concentration. What that means is, think of like you're pushing a big rock uphill, or you're trying to get into a crowded subway bus or subway train. So you're going from an area of low concentration to high concentration, and to do that would require energy. Thanks. So active transport processes accumulate necessary substances on one side of this cell membrane at concentrations many times greater than those on the other side. The major example of active transport that we'll talk about is the Sodium-Potassium Pump. And it's kind of exactly what its name says it does. It will move sodium out of the cell and potassium into the cell. And it's a pump, it requires energy, and the result will always be a higher concentration of sodium outside cells and higher concentration of potassium inside cells. Sodium is Na positive and potassium is the K positive. So that's the sodium-potassium pump. It just takes two substances and flip-flops them into and out of the cell. Here's a look in their cell membrane. Remember, it's a lipid bilayer, so it's made of these phospholipids with the little heads and the tails facing in. And here's our pump, and here's a little spot for ATP, which is energy to bind. So when ATP will bind to this pump, it will pump sodium out of the cell, and it will pump potassium into the cell. And again, sodium is abbreviated Na in the periodic table, and potassium is abbreviated K. They're both positively charged, and because they're positively charged and moving against their concentration gradient, they need a pump or a carrier to move them across the cell membrane. Secondary active transport also uses energy by a concentration gradient established by the active transport of one substance, such as sodium, to transport other substances. So we call it secondary active transport because it'll use the energy provided by the concentration gradient of one substance to move something else. And no additional energy is required above that. In co-transport, the diffusing substance moves in the same direction as the initial active transported substance. So in counter-transport, they'll move in opposite directions. So in co-transport, they'll move in together, and in counter-transport, they'll move opposite to that of the initial active transported substance. So co-transport moving together, counter-transport moving opposite that each other. And again, we're talking about secondary active transport, which just uses the energy established by the concentration gradient in one substance to move something else. I know that's kind of confusing. Let's take a look at this picture here. So for example, this is secondary active transport. This is showing counter transport because sodium is being pumped out when potassium pumps in. And here, number 2, this is co-transport. While as sodium comes in, glucose comes in with it at the same time. So counter transport is opposite directions, and co-transport is same directions. Hopefully that kind of makes sense. Endocytosis is a type of active transport, so it requires energy. And endo, the prefix endo means inside. So endocytosis is a process that brings materials into cells using vesicles. And a vesicle is, again, just a container. We have different types of endocytosis. Really briefly, receptor-mediated endocytosis happens when a substance will bind to a receptor molecule on the cell membrane and then get transported in. And then we have phagocytosis and pinocytosis. Phagocytosis means cell eating. So this will be for larger solid particles when they're ingested. And penocytosis means cell drinking. So penocytosis is for smaller vesicles, and they will always contain liquid rather than solid. So phagocytosis and penocytosis are two ways that the cell brings substances inside. And again, active transport energy is required. Phagocytosis, cell eating, so these will be solid, larger particles. And penocytosis, cell drinking, smaller liquid particles. This is just a look at receptor-mediated endocytosis, showing how the molecules being transported will bind to receptor molecules on the cell membrane. And you can see here how the cell membrane kind of starts to fold in on itself, and it creates a vesicle to get these molecules inside the cell. So it's really kind of this artistic process that the cell, not artistic, but really an interesting process how the cell gets things inside of it. So that's receptor mediated. And then exocytosis. So endocytosis, endo, inside, exocytosis, exo, exit. So this is bringing things outside of the cell. So it involves the use of the membrane-bound sacs called secretory vesicles that accumulate materials for release from the cell to the exterior. These vesicles will move to your cell membranes, they'll fuse, and then they'll release their material by exocytosis. And an example are the secretion of your digestive enzymes in your digestive tract. But let's just take a look at a picture of what exocytosis is. So if we start at number 1, here's our container, our vesicle, containing the contents that the cell wants to get rid of. These could be digestive enzymes. It could be a waste that the cell wants to get rid of. So this vesicle will move up to the cell membrane, fuse together with the cell membrane, and spit out or vomit its contents to the exterior of the cell. So this is, again, exocytosis is things exiting the cell. This is a really cool, this is transmission electron microscopy. That's what TEM stands for. It's at 30,000 times magnified, actually showing exocytosis occurring with this vesicle fusing together with the cell membrane and the contents being spit out. So kind of cool there. Questions so far? Again, these are all forms of active transport, so they require energy. Now we'll get into the general cell structure. And we'll go over this, well, not quickly, but this won't involve any sort of processes. We're just going to be naming things inside the cell and what they do. So the interior of every cell is composed of cytoplasm. It's a jelly-like fluid that surrounds the organelles. Think of kind of like Jell-O or strawberry jelly. And organelles are tiny organs. They are specialized structures that perform certain functions. And these are the different types of organelles that we are going to go over right now. So again, we're looking at the inside of the cell. We talked a whole lot about the cell membrane, and now we're looking at the inside of the cell at all of these organelles. The cell nucleus, we'll start with the cell nucleus. Yep, we're on chapter 3. Oh, thanks guys for answering too. So the cell nucleus, it's a large organelle. It's usually located near the center of the cell. Think of this as the brain to the cell. It's bounded by its own nuclear envelope or its own kind of membrane, which consists of outer and inner membranes with a space in between. And the nuclear membrane contains pores or openings that just allow certain things to go into and out of the nucleus. The nucleus of human cells contains 23 pairs of chromosomes, and these just house the DNA and the proteins. And the DNA are all of the genetic materials of the cell. During most of a cell's life, the chromosomes are loosely coiled and collectively called chromatin. So chromatin is just the loosely coiled version of DNA in the nucleus. But when a cell is getting ready to divide and reproduce, these chromosomes will become tightly coiled and they'll be visible underneath a microscope. Within the nucleus are nucleoli, and these are, diffuse bodies with no surrounding membrane. There are usually 1 to several nucleoli, and the subunits of ribosomes are formed within the nucleolus. And the nucleolus is another central part within the nucleus. These ribosomal components that are created in the nucleolus will exit the nucleus through nuclear pores. So subunits of ribosomes. Ribosomes are used to create proteins, and the parts needed to make up the ribosome itself are formed within the nucleolus. So here's a look at the cell nucleus, and you can see the nucleolus is kind of the center part of the nucleus itself. Here's the nuclear envelope, which is just made of 2 layers with a space in between. You'll see pores that are openings through that envelope that'll allow things to go into and out of it. And then you'll see ribosomes attached kind of to the outside of it. So this is the cell nucleus. Chromosome structure, again, your DNA is within your nucleus. And right before your cells divide, your DNA will become tightly coiled or wound around these proteins. And they will become tightly coiled to form this X-like structure, which is a chromosome. So a chromosome is just DNA wound up really tightly together. It's found in the nucleus of the cell, right before your cells will divide. Okay, so ribosomes. Ribosomes, like we said, their components are produced in the nucleolus of the nucleus. And the ribosomes, when I say ribosome, I want you to think protein. Ribosomes make proteins. They might be attached to other organelles like the endoplasmic reticulum that we'll see, or they might just be free floating through the cell. So ribosomes create proteins. They'll be attached to another organelle, or they'll just be free floating in the cell. But ribosomes make proteins. Here's a look at how ribosomes are produced. This is just showing within the nucleus of the cell. It's showing the different components of a ribosome that are created, and then they exit out of the nuclear pores and they come together to make a ribosome. So the parts of the ribosome are created inside the nucleus, and then they'll exit out of the nucleus and become a ribosome itself. This is showing a strand of mRNA. That just has to do with when your DNA replicates itself before it divides. It'll use the ribosome for help in doing that. So ribosomes are produced in the nucleus. They leave the nucleus and they're used to make proteins. Endoplasmic reticulum. It's a series of sacs or flattened tubes that extends from the nucleus, that nuclear membrane, into your cytoplasm. The endoplasmic reticulum, we often abbreviate it ER, and it can be rough and it can be smooth. And it's really easy to determine what rough means and what smooth means. Rough means that ribosomes are directly attached to the endoplasmic reticulum, and the rough ER will be involved in protein synthesis because ribosomes create proteins, and the rough ER has ribosomes attached to it. The smooth ER has no attached ribosomes. Your smooth ER is the site for lipid or fat synthesis, cellular detoxification, and it also stores calcium ions in skeletal muscle. Your smooth ER does a lot of cellular detoxification. So remember I said at the beginning of all this, when we're talking about our cells, your cells might have different numbers of organelles if they're performing a certain purpose. So you'll find a lot of cells with smooth ER in your liver. Why do you think that is? Why would your liver have a lot of cells with smooth ER? What does your liver do?
Filtrate.
It filtrates and it detoxifies. So yeah, filters, but yeah, detoxifies everything harmful you take in, whether that's a drug, alcohol, it all goes to your liver. That's why alcoholics, chronic drug users, they got to be very careful because they're really damaging their livers. But smooth ER will be found in cells in your liver because that helps with detoxifying some of those harmful substances. So here's what endoplasmic reticulum. Remember, it'll be surrounding or located close to the nucleus because the nucleus creates the ribosomes that will become attached to the rough ER. And eventually the rough ER will just change to smooth ER without ribosomes. But again, the endoplasmic reticulum is often found right next to your nucleus because it's taking those ribosomes that the nucleus formed and they're being attached to the rough ER. Then we get to the Golgi apparatus, sometimes called the Golgi complex or just the Golgi. It also consists of closely packed stacks of curved membrane-bound sacs or flattened tubules. Your Golgi apparatus collects, modifies, packages, and distributes proteins and lipids manufactured by your endoplasmic reticulum. Remember, the rough ER had to do with creating proteins, and the smooth ER helped to create lipids. When lipids or proteins are made by the ER, they get sent off to the Golgi, and the Golgi collects those lipids or proteins, it modifies them, it puts them in a nice little package, and then it distributes them. So I compare your Golgi apparatus to an Amazon distribution center or an Amazon plant. So, you know, we buy things on Amazon all the time, and it comes from something like the Golgi. So your Golgi apparatus will form vesicles or containers that have these proteins and lipids inside of them. Some of them will be secretory vesicles, which will leave the cell. Others might be lysosomes, and we'll talk about what lysosomes do, or some sort of other vesicle. So here's a look at what the Golgi looks like. You can see how things will come in this side of the Golgi, and then little vesicles will be kind of pinched off and sent either to the cell membrane, where they will leave through exocytosis, or they might stay within the cell if the cell needs different proteins to function. So that's your Golgi apparatus. Lysosomes are something that the Golgi apparatus creates, and lysosomes contain lots of enzymes that function as intracellular, meaning they will function within the cell, digestive systems. So your lysosomes are like the garbage truck of the cell. Your lysosomes go around to the inside of the cell and help to digest all the waste. So lysosomes, they're formed from your Golgi. They have a lot of enzymes in them that help to break stuff down or digest it, and they will be intracellular, or they'll stay within the cell. Vesicles formed by endocytosis coming into the cell might fuse with lysosomes first in order to break down what's inside of them. And one example of this are your white blood cells, and they will phagocytize bacteria. So if bacteria enters your cells, these lysosomes will take in that bacteria and try to digest it before it can harm anything within your cell. So here's a look at lysosome action. A vesicle will come into the cell, maybe with something harmful inside of it, or just pieces of it that need to be digested or broken down. And the lysosome that's coming off the Golgi will fuse with that vesicle and break down whatever is inside of it. So that's what lysosomes do. They digest harmful things or just waste products within the cell. Is everyone with me so far? Questions? I know we're going through a lot of material here, but at least hopefully it makes more sense than the transport part. Okay. Peroxisomes or perioxisomes, these are also small membrane-bound vesicles. They contain enzymes that break down fatty acids, amino acids, and hydrogen peroxide. So peroxisomes are like lysosomes, but they break down specific substances. They break down fatty acids, amino acids, and hydrogen peroxide. This is important because hydrogen peroxide is a byproduct of fatty acid and amino acid breakdown. It can be toxic to a cell. So we need these peroxisomes to get rid of a toxic substance, which is hydrogen peroxide. So they will break down hydrogen, which again can be toxic to the cell itself. Mitochondria. So mitochondria is an important one. Mitochondria create all of your cells and bodies ATP. So that's the big thing about mitochondria. If you leave today's lecture with anything, if you can remember that mitochondria create ATP and ribosomes create proteins. Just start with those two and just try to remember those two today and learn another couple organelles tomorrow. So mitochondria, they're singular if you say mitochondrion. They're small organelles and they create ATP by aerobic metabolism. That means that you need oxygen to create energy in your cells, and creating the ATP happens in your mitochondria. They have two membranes. These membranes are folded on top of each other, and we call them cristae. And I'll show you a picture of what that looks like. The material within the inner membrane is the mitochondrial matrix. This will contain enzymes and mitochondrial DNA, which is its own separate genetic material for mitochondria. And cells with a large energy requirement have more mitochondria than cells that require less energy. So in your body, think maybe anywhere in your body, what kind of cells do you think would have a lot of mitochondria?
Muscle?
Muscle. All of your muscle cells have tons of mitochondria, especially your cardiac muscle that's constantly beating. Even the muscles in your back, which are helping us sit up straight. All of us probably have really bad posture right now. You have lots of mitochondria in your muscles because your muscles constantly need ATP to function. So here's a look at what a mitochondria looks like. Again, it has this double membrane, the inner and outer membrane with an intermembrane space in between. And then we call the matrix that inside space. I brought up the word cristae or crista. Those are just the folds that the inner membrane makes. Mitochondria are fun to look at underneath a microscope. Again, one that's much more powerful than we would have in lab. But you can see these folds and the double membrane of these mitochondria in the cell. So mitochondria create ATP. Big picture, that's what you should know about mitochondria. They create ATP. The cytoskeleton, so just like we have a skeleton and bones that gives us our shape and holds our framework up, your cells also have a cytoskeleton. The prefix cyto means cell, and this gives internal framework to the cell. It will consist of different proteins that support the cell, hold all these organelles in place, and help the cell to change its shape. And these protein structures will be microtubules, microfilaments, and intermediate filaments. And think of these different types of tubules and filaments. They're just the cell's bones. So they give the cell its shape and hold everything up. So we'll go over these three types. Microtubules will be hollow. They'll be formed from protein subunits. They have a variety of roles. They'll help assist in cell division, and they also will help to form cilia and flagella. Flagella help cells move, and the only cell in the human body that has a flagella or a tail, what do you think is that? The sperm cell. The sperm cell is the only cell in the human body that has a flagella. Cilia will be attached to kind of the exposed surface on some epithelia, and cilia will help move mucus through your respiratory tract. I think we talked a little bit about that. Okay. Microfilaments, they're a little bit smaller. They help support the cytoplasm, giving the cell its shape. Microfilaments are involved with muscle cells to help muscles shorten or contract. So two types of microfilaments would be actin and myosin, and those just help your muscle cells to shorten or contract to help provide movement. Then intermediate filaments are the third type. They provide mechanical support to the cell, and a specific type of intermediate filament is keratin, which we've talked a little bit about. And that's a protein associated with your skin cells to provide protection. So these are the cytoskeleton and what they look like. Here are microtubules. They have the largest diameter. Here are intermediate filaments. And here are the microfilaments, have the smallest diameter. And again, each one of these cytoskeleton pieces have a slightly different function, but in general, they all work to help give the cell its shape and support the cell. Questions, concerns? Am I moving too fast?
What's the measurement on the side, like 25 nm?
So NM is a nanometer.
Okay.
So nano, what is nano? 10 to the negative, is it 9?
Bajillionth? I don't know.
Does anybody know? Yeah, extremely small. On the power of 10 to like the negative 9 or something. Negative 6 or 9. If anyone knows that, they can correct me. But it's a nanometer, extremely small. Good question. Other questions? Okay, so centrioles, we'll show a picture of what they look like in a second. The centrioles are important for mitosis, which is cell division. The centrosome is a specialized area of your cytoplasm close to the nucleus where microtubule formation occurs. And the centrosome contains 2 centrioles. So centrioles always come in a pair of 2, and we call that pair a centrosome. They're normally oriented perpendicular to each other like that. So 2 centrioles are formated kind of like an X, and that makes up 1 centrosome. They're cylindrical, they're made up of little microtubules, and they will help in mitosis to pull things apart from the cell during cell division. Nanometer is 1 billionth. So how many zeros is that? 9, right? Yeah, so it would be 10 to the negative 9. Thanks, Joshua. So here is 1 centriole, and you can see how it's kind of made up of these long tubes that are put together. If you go back to the first picture of the cell, you would see how the 2 centrioles will be perpendicular to each other. But this is what a centriole looks like, and they will help to pull chromosomes apart during cell division. Cilia then, these are projections from the surface of certain cells. We talked about cilia when we talked about epithelium in your respiratory tract. And specifically cilia there will move mucus. They're cylindrical structures that extend from the cell. They're also composed of microtubules. Flagella have a structure similar to that of cilia, but they're much longer, and they usually occur in only just 1 per cell. So sperm cells, they're the only human body cell that has a flagella, flagellum for singular, and that propels the sperm cell. Microvilli then, these are specialized extensions of the cell membrane. They're also supported by microfilaments. They don't actively move. They more help to increase surface area to help with digestion. So microvilli just help to increase surface area of the tissue that they're on. So you'll find them in your intestine, your kidney, and other areas in the body in which absorption of something is extremely important. So those are microvilli. The whole cell's activity, like we were kind of talking about, a cell's characteristics are determined by the type of proteins that it produces. And the proteins produced within that cell will are in turn determined by the genetic information in the nucleus. So the nucleus with the DNA contains kind of the instruction code for what proteins to be produced, and that will guide or tell the cell what type of cell to be and what to do. So information in DNA will provide the cell with a code for its cellular processes, and DNA will provide this code in the form of protein. So what is DNA? And I just want to double check where we are here. Okay. Any questions before we start with DNA? We'll go through the DNA a little bit slower because this will be a little trickier. Okay. Just wanted to see where we were in the PowerPoint. All right, so DNA contains all the information that directs protein synthesis. And creating proteins from DNA, we call that gene expression because the genetic material in DNA will be expressed in the form of a protein that it makes. A DNA molecule consists of nucleotides, which are joined together to form 2 nucleotide strands. And these 2 strands are connected and resemble a ladder that is twisted around an axis. So we get this helical pattern, like a ladder twisted on itself. And each nucleotide consists of a 5-carbon sugar, a phosphate group, and a nitrogenous base. Each nucleotide on one DNA strand has a specific bonding pattern to another nucleotide on the opposite strand. So the bases will only attach to another specific base, and those bases will attach to each other through hydrogen bonding. A gene, when we talk about your genes in the human body, it's just a sequence of nucleotides that provide a chemical set of instructions for making a specific protein. So that's what DNA is. It's filled up with genes that provide instructions for making proteins. Expressing these genes is just another word for saying protein synthesis. So when we say we're going to talk about gene expression, all we're doing is we're creating proteins, because expressing those genes, those nucleotides in the DNA, means we're creating a protein from it. So we're following the instructions in the DNA to create a protein. This process involves transcription and translation. So to create a protein, we need to transcribe and then we need to translate. Transcription involves copying the DNA into what we call messenger RNA. So the DNA will need to be copied to another form first, called messenger RNA. And then translation is the process that involves that messenger RNA, which was the copied instruction from the DNA, being used to produce a protein. So transcription is copying DNA into messenger RNA, and translation then is just using the code or those instructions in the messenger RNA to make a protein. So we'll talk about transcription first. And you should know that transcription occurs in the nucleus of the cell. So transcription occurs in the nucleus of the cell. And again, transcription is when you'll have your double strands of a DNA segment that'll separate. Then the DNA nucleotides of the gene pair will pair with RNA nucleotides that form the mRNA. So I want to show you a picture here. Okay, we'll finish talking about transcription and then I'll show you a picture of what I mean by this. But transcription occurs in the nucleus, and during transcription, the double helical kind of ladder of DNA will just separate, and mRNA will line up with the separated strands of the DNA. DNA contains one of the following organic bases: thymine, adenine, cytosine, and guanine. And these bases will bind the two strands of DNA together. Messenger RNA contains all the same bases except uracil will replace thymine. So messenger RNA contains uracil, adenine, cytosine, or guanine. And DNA nucleotides only pair with specific RNA nucleotides. So when we've separated out our two DNA strands, RNA will make up the second half of those newly separated strands. The thymine will pair with RNA's adenine. DNA's adenine will pair with RNA's uracil. Cytosine and guanine will always pair with each other. So what does all this mean? You're probably lost. So this is a strand of DNA, and these nucleotides here are shown. Cytosine will pair with guanine. That's the yellow and green the base. So when I talk about bases, these are bases of the nucleotides that can only pair or bond to each other. It's important for these to specifically bond with only the same one that they're allowed to, because they form a hydrogen bond. That's the weakest type of bond. When these bases form, they allow the DNA to be wound. So this is DNA being wound with its bases pairing with each other. Cytosine will pair with guanine, and thymine will pair with adenine. During transcription, these two strands of DNA separate. So they'll break apart from each other. So those hydrogen bonds that are holding those bases together break apart. When they break apart, the mRNA strand comes in and forms the other half of the newly separated DNA strands. So we have this DNA strand breaking apart, and we have the messenger RNA strand coming up and forming the other half of that newly separated out DNA. When we form the mRNA, the only difference One of the only differences is in the base pairs. When you're forming mRNA with DNA, you don't have thymine anymore. Thymine has been replaced with uracil. So now when we pair with adenine, we'll get a uracil on the messenger RNA strand. This probably goes into even more detail than what you guys will be tested on. You should just know that transcription is separating out the DNA strands and making a messenger RNA strand to be the first step in gene expression or protein synthesis. And again, this occurs, where does this occur? Where does transcription occur?
The nucleus itself.
The nucleus, good. Any questions on this before we go on to translation? On a scale of 1 to 10, 10 being utterly confused, 1 being this is okay, just write your number in the chat. And I'm going to keep going, but scale of 1 to 10, 10 being utterly confused, 1 being I'm okay. Okay, so not many 10s. 5-ish, that's fine. You can be honest too. Okay. Okay. So transcription occurs in the nucleus. What we've done in transcription, we've separated out DNA. Again, we're making an instruction for a protein to be formed, and we've copied that DNA onto the mRNA. So translation now will occur in the cell cytoplasm. So after we create that mRNA strand that mirrors the DNA, that now double-stranded structure actually leaves the nucleus through the nuclear pores. So now we're going out into the cytoplasm, that jelly-like substance. Now we get to kind of a fun part, easy, I shouldn't say easy. The messenger RNA strand that we just formed from DNA will now attach to a ribosome. What do ribosomes do?
Protein.
Ribosomes make proteins. And again, gene expression is making proteins. So eventually we got to get to a ribosome because their ribosomes will create the protein. They were just telling me how confused you were, Asil. So 10 if you're utterly confused, or 1 if you're not confused at all. So I was just asking for a poll. So translation then, the mRNA will attach to a ribosome, and a codon on the mRNA will be read by an anticodon on the transfer RNA to create a protein. Hopefully we won't get into too much detail here. You guys don't need to know all this. But translation, the transfer RNA will transport specific amino acids that the messenger RNA is coding for from the cytoplasm to that ribosome mRNA complex, and it'll initiate formation of the polypeptide chain. Polypeptide just means many amino acids in a line to create a protein, and the process will continue until the entire polypeptide or protein will be formed. So this is translation of mRNA. So we see our strand, let's go with, okay, we'll start up here with number 1. This is the ribosome. There's like a large and small subunit to the ribosome, and this is our mRNA strand that kind of gets shuffled through the ribosome like a conveyor belt. And there's 3 base pairs, which are codons, and they will code for a specific amino acid that a transfer RNA molecule brings. And the transfer RNA molecule is this little green or orange-like structure. The little green circle is the amino acid that it brings, and it has to line up with the specific code on the mRNA strand. So the mRNA strand kind of gets fed through this ribosome like a conveyor belt, and every 3 base pairs is a different code for a different amino acid that the transfer RNA can read, and it brings these amino acids as the mRNA strand gets fed through the ribosome, and we start to create a protein. And we call this a polypeptide chain when we bring in several amino acids together and we start to form a long chain. And that's our protein synthesis. So this is a good, maybe this is probably the best overview of gene expression to get not bogged down by details. Your DNA contains the information necessary to produce proteins. So that will be in the nucleus. Transcription of one DNA strand results in messenger RNA, shown right here where my cursor is. That's just a complementary copy of the information on DNA. That mRNA strand leaves the nucleus and heads to a ribosome, and the mRNA gets fed through the ribosome. Amino acids, which are the building blocks of proteins, are carried to the ribosomes by transfer tRNA molecules. And in the process of translation, the information contained in mRNA is used to determine the number, the kinds, and the arrangement of amino acids in that polypeptide or protein chain. So this is gene expression, also known as protein synthesis. Okay, before we go into cell cycle, questions on this? I should have waited to ask the confusion level till now, maybe. Well, let's talk about something else. So the cell cycle, well, it's all really the same thing, but the cell cycle is just the growth and development of the cell that happens right before it divides. So during growth and development, cell division occurs to increase the number of cells or replace damaged or dying ones. So from the time you're born, your cells are dying. So that's why our lifespans, we're always on the way to the grave because all our cells are constantly dying. So our cell cycle, that was a really kind of sad topic. I didn't mean to say that. That came out worse than it did. I meant to. But the cell cycle division, the cell cycle includes division and growth, and it includes the two major phases, a non-dividing phase called interphase, and that's just when the cell will grow, and the cell dividing phase, and that is called mitosis. So we're going to talk about this. Yeah, it was dark, sorry. So the cell spends most of its life cycle in interphase, and this is just when it performs its normal functions. So most of your cells' lives, they're just performing their normal functions. If that's creating a lot of mitochondria in your muscle cells, they're doing that. If it's detoxifying what you ingested over the weekend, they're in your liver doing that. So that's what your cells normally do. During interphase, the DNA will be replicated because in order for your cells to divide, they need to replicate their DNA, their genetic instructional material, so that each new daughter cell that's formed will have DNA. So during interphase, the two strands of DNA just will separate from each other, and each strand serves as a template for production of a new strand of DNA. So interphase, your DNA, one single strand, will replicate itself to form two new strands of DNA. Nucleotides in the DNA of each template strand will pair with new nucleotides that will be joined by enzymes to form a new strand of DNA. So this is just DNA replication. Replication of DNA gives two identical chromatids that are joined at a central centromere, and both of these will form one chromosome. So this is a look at DNA replication. How DNA replicates itself is kind of similar to what we talked about in transcription. The DNA strands will separate, and new DNA strands will come up and join those strands that have separated out. So at the end, you'll get two new DNA molecules. Yes.
At this point, though, they're still in one cell, right?
Yeah, they're still in one cell. The cell is not divided yet. Yep. So this occurs during interphase, normal cell functions. The DNA will replicate itself, still being in one cell. All of your human cells, except sperm and egg, contain 23 pairs of chromosomes for a total of 46. The sperm and egg will contain 23 chromosomes total, and that's because the goal of the sperm and the egg is joined together to form a new zygote or fetus that would contain those 46 chromosomes. But each human cell has 23 pairs of chromosomes for a total of 46. One pair of chromosomes are the sex chromosomes, which consist of 2X if the person is female, or an XY if the person is male. And then we'll talk about mitosis. So mitosis is the other part of the cell cycle. So interphase is normal stuff that the cell does. It replicates its DNA. It handles all the normal functions. Mitosis makes up like less than 10% of the cell cycle. And mitosis involves forming two daughter cells from a single parent cell. Mitosis will be divided into 4 phases: prophase, metaphase, anaphase, and telophase. And if you can remember the acronym PMAT for the order that these phases will go in. So PMAT, PMAT, give you the correct order of these mitosis phases. So now we'll get into how the cell divides. During prophase, your chromatin, which contains your DNA, it will condense to form chromosomes. Microtubules will form to assist in breaking up the centromere. And again, the centromere was the center part of each chromosome to help it divide or separate. And the nuclear membrane of the cell dissolves because we're going to be separating the cell into 2. We got to get rid of that nuclear membrane first. So in prophase, the chromosomes are forming, and the microtubules are also helping to form to assist in breaking them apart. So that's prophase. Metaphase, the chromosomes line up near the center of the cell. The movement of the chromosomes will be regulated by the attached spindle fibers. So I'll show you a picture of what I mean by this. But during metaphase, basically if you have a circular cell, in metaphase, all of your chromosomes line up in a line through the center of the cell, and it's kind of fun to see in a microscope. Then during anaphase, your chromosomes will just be pulled apart. So your chromatids will separate, and each chromatid is called a chromosome, and each of the 2 sets of chromosomes will be moved towards opposite sides of the cell. So anaphase, under a microscope, you can see these chromosomes being pulled apart. And I wish I could show you there, we'll get to a picture here. And then telophase, the chromosomes in each of the 2 new daughter cells become organized to form 2 new separate nuclei, or a nucleus, one in each newly formed daughter cell. The chromosomes will begin to unravel and resemble what normal genetic material looks like. And following telophase, the cytoplasm will divide completely to create these 2 separate daughter cells. So this is a look at the cell cycle. Interphase, again, is normal growth, normal development of the cell, normal processes. In prophase of mitosis, we start to see chromosomes forming. And chromosomes are made of 2 chromatids in this X-like structure, and the centromere refers to just a center piece that's holding them together. So during prophase, the nuclear envelope will also start to dissolve because these chromosomes eventually need to get out of the nucleus. And again, a chromosome is just the cell's DNA tightly wound up. So the DNA is just getting ready to be moved and an easier function than just being all over the place. So that's why chromosomes exist. Centrioles will start to move to opposite sides of the cell. And in metaphase, we get a nice lining up of all of these chromosomes. And again, I think it's fun. Underneath a microscope, you can see the different stages that a cell is in, and metaphase is really easy to spot because all the chromosomes are nicely lined up along the center of the cell. You'll also start to see what we call spindle fibers, which are just fibers coming off of these centrioles, and those will attach to the chromosomes to get ready to pull them apart. And they will pull them apart during anaphase. So again, under a microscope, we can actually see these chromosomes being pulled apart, and you can see the spindle fibers reaching out and touching them to pull them apart. So again, we've replicated our DNA, we've lined up the chromosomes, and now we're pulling them apart during anaphase. In telophase, you start to see a new nucleus form in each new daughter cell. The cell is technically still connected. You might see an indentation start to appear where the cell is about to divide its cytoplasm completely. But in telophase, we just start to see new nuclei form. And then mitosis is complete in what we call cytokinesis. And that just means kinesis means, well, cytokinese, split or separating out the 2 new daughter cells. So you'll see a new wall begin to appear between the 2 new daughter cells. So that's the cell cycle with that. Questions on the cell cycle?
I actually have just kind of like a random question.
Mm-hmm.
When you teach this class in, physically teach this class, is it a little bit easier to understand and is it more hands-on kind of thing?
Yeah, I would say like if I were I would probably be drawing these pictures up and we'd going through one at a time. And also during lab, you would be looking at these cells under a microscope and you would try to pick out, oh, this cell must be in metaphase. This cell must be in anaphase. So yeah, I would say if we were in person, it would probably be a little easier to understand like most everything.
All right, thank you.
Mm-hmm. Hopefully we go back to person someday. Yeah. Any other questions about the cell cycle? So what were the stages of mitosis in order? What comes first?
Prophase,
Prophase and then
metaphase,
anaphase,
anaphase.
and telophase. Good. Then interphase, what does the cell spend most of its time in? Mitosis or interphase?
Interphase,
Interphase. So mitosis only occurs when the cell needs to replicate itself to make up for cells that have died or don't work anymore. Good. Okay, differentiation. You guys are doing great and we're getting close to being done with this chapter. Differentiation is just the process by which cells will develop into their own specialized structure and function. So when a sperm cell and an oocyte, which is an egg, unite to form a single cell, then all of a sudden a great number of mitotic divisions occur to give the trillion cells of the body. So if you think about it, it's pretty amazing that a sperm and an egg come together to form one single cell, and immediately they start to divide into more cells that will go on and differentiate. The process of differentiation means that those cells will go on and to continue to form the specialized structures they need to do their function. During differentiation of a cell, some portions of DNA are active, but others will be inactive because DNA contains the set of proteins required for a function, so we'll only have the active portions that are needed specifically for that protein. So this is kind of fun to look at, a little more macroscopic, so it's easier to understand, but the diversity of cell types out of one single uniting of a sperm and an egg will get differentiation into bone cells, nerve cells, muscle cells, and red blood cells. So it's pretty amazing that all of our cells just came from the uniting of two cells into one, and then we get all of this diversity from that. We'll go over what these different cells will look like, I think when we get to those chapters. Apoptosis. Yep.
I'm sorry, I'm taking a step back here, but I had a question about interphase and mitosis. You said that interphase occurs normally, like that's when the cells are operating and functioning properly, and mitosis occurs when cells die. Does that include like basic injuries to the human body when they die? Like not, because you made the example of a human dying, but cells die typically, like skin cells die on a normal basis.
Right, and all your cells, I more meant to say, I know that came out wrong. I more meant to say all of your cells are under a timeline. They won't live forever. So all cells will die eventually. So mitosis is needed for cells to like replicate themselves and reproduce new cells to make up for the cells that have died off, for cells that have been injured, like you said, cells that are overcome with disease or bacteria. Cancer cells divide constantly, and they continually divide, and we don't know how to stop them from dividing. So that's why cancer is so scary, because they constantly divide and spread. Does that help answer your question?
Yeah, very much so. Sorry, I was just a little confused as far as like the human body versus the cell itself.
Yeah, sorry about that. That was, I meant, yeah.
Would you say the cell cycle is going on 24/7?
Oh yeah. So most of your cells right now are in interphase because what are they doing? They're making energy. They're helping contract your muscles. They're beating your heart. So most of them are just in interphase. They're growing. They could be replicating their DNA to get it ready for mitosis, but interphase is just normal cell carrying on its normal processes. But also at the same time, you have cells in your body that are undergoing mitosis and cells that are in each stage of mitosis. So it's really fun to look at a section of cells underneath a microscope, and you can see cells in each stage. You'll see a lot of cells in interphase, normal cell structure with a nucleus. You'll see some cells in prophase, metaphase, anaphase, and telophase. But yeah, think of all the trillions of cells in your body. They're in one of these stages.
Hey, Professor, does the genes change on the cancer cell?
Yeah, so there are, I think, particular genes that they found that could go on and lead to cancer cells. Think of like breast cancer, the BRCA gene that they've been able to figure out through genetic testing. Because what makes up cells? Proteins make up cells, and proteins are made from DNA. So within the DNA, if you have something off, even in a base pair, we call that a mutation. If one of the bases is mutated, that will go on and completely mutate a particular protein, which could affect a cell or a function. So yeah, it all comes back. That's why DNA is so important, because how those base pairs are lined up and the sequence of them. Yeah, they say there might be, if you say something is inherited or a genetic defect, it's because that portion of your DNA was passed on through a sperm or an egg, and you got it when the sperm and the egg united. It can be a recessive or a dominant trait. My
Is it
daughter,
possible to target that specific gene on a human body?
yeah, they're talking about genetic therapy, or, I mean, I'm sure if any of you have had kids, you've probably been through genetic testing. So you can know off the bat if the fetus inside of you might have a defect, or you would have passed on, if they know your genetic history, you might have passed that on. So in terms of targeting the gene, I don't know if they can go in right away and like splice it open. I mean, in terms of doing surgery at a genetic level, I think there's a lot of work in genetics. And if you guys are interested in that, I would encourage you to go on and take more science, because it's really fascinating stuff. That's really where healthcare and medicine is right now. It's all in genetic therapy.
I heard that genetic test, though, when you're pregnant, isn't really mostly accurate. It can be flawed a lot.
Yeah, it can be.
Okay.
I didn't do it at all because I didn't really care. But I think it's more so if you like for a peace of mind. But yeah, it could be flawed. Yes.
Well, it's just our bodies are so crazy. Like just like a baby even inheriting, like just swallowing poop on the way out. Like let's say they took too long to come out, and then they poop inside the belly, and then they swallow poop. That can make the baby go, have mental health. It's just so crazy. Our body is so crazy.
I know, it's crazy.
Real quick, just to clear something up. So cell division starts with transcription when the DNA starts to separate. Is that like the initial point?
Are you talking about gene expression or the cell cycle?
I'm probably combining the two.
That's okay. Yeah, because they're kind of so like DNA replicating itself happens during the cell cycle, because DNA has to make you need two identical strands of DNA to be passed on to two new cells. So that's part of the cell cycle. Transcription was DNA separating, but messenger RNA, which is a new strand that forms with those genetic instructions, that's involved to create proteins.
Okay, all right.
Yeah, and that's gene expression. Yeah, it's a lot of information. Yep. Any other questions? Yeah, it's really fun to talk about recessive and dominant traits. My daughter has red hair. My husband and I do not have red hair, but it's recessive, so it must have come somewhere back the line on both of our sides. So that whole idea too is what your kids look like. I don't think our kids look anything like us, but I know they're ours. But
yeah, I'm waiting on my 5-month-old. She looked like she has like maybe like green or blue eyes, and none of our family got any. So I'm like,
wow. Okay.
What's going on?
Yeah, it's fun. That's crazy though. All right, apoptosis, more about cell death. Hopefully this won't become too dark. But apoptosis, all cells can also program themselves to die. And this is a normal process by which cell numbers within various tissues will be adjusted or controlled. In the developing fetus, apoptosis will remove extra tissue, such as cells between the developing fingers and toes. So you might have heard of people being born with webbed toes, which is just extra tissue between their toes. There was something wrong. Apoptosis wasn't occurring to remove that extra tissue when they were developing. In some adult tissues, apoptosis will eliminate excess cells to maintain a constant number of cells within the tissue. So kind of this idea of homeostasis and regulating things, apoptosis helps to keep that cell number consistent. Cellular aspects of aging. These are various causes for cellular aging: existence of the cellular clock. That just means that all cells will not last forever. The presence of death genes. I'm not really sure what that means. A death gene or a disease that would speed up. I'm not sure what that means. DNA damage. So we talked about mutations. Damage to DNA can come from UV rays from the sun. That can cause skin cancer. Damage can be something else external. If you think about the Chernobyl explosion, or when America dropped the bomb on the Japanese in World War II, I mean, the people who were affected by that had damage to their DNA that affected them. Formation of free radicals that aren't able to be taken care of, and mitochondrial damage. A lot of mental defects in kids, or just really in anyone, that occur during development actually damage parts of the mitochondria. Why is it bad if we damage mitochondria?
No power supply.
Energy.
No energy and no power supply. So, I mean, there's specific pieces of the mitochondria that certain toxins, certain poisons will go and damage specific parts of the mitochondrial chain of creating ATP. When those are damaged, if the cell doesn't have energy, that's how you die. So a lot of toxins and poisons actually damage mitochondria. Tumors are an abnormal proliferation of cells. They can be malignant or benign, which is non-cancerous. So this proliferation of cells that's uncontrollable, they're due to problems occurring in the cell cycle. Some can be benign and some can be malignant, which means they're cancerous. Malignant cancerous tumors can spread by a process, and we call that metastasis. So if you hear of cancer metastasizing, that just means the tumors or those cells are spreading. Uncontrolled cell growth is cancer, and uncontrolled cell proliferation. And I guess that's it. Let me stop the recording